Welcome to Diagnosing Dx, the podcast giving diagnostics the platform it deserves. While therapeutics and devices often take centre stage, diagnostics is the engine driving precision medicine. I'm your host, David Philby, and each week we spotlight the innovators, breakthroughs, and real-world challenges shaping the future of DX. Arnon Chait, founder of Cleveland Diagnostics, breaks down how they took a flawed but widely used test like PSA and rebuilt it into something that can genuinely change clinical decisions. We also get into why most diagnostics fail, what people consistently underestimate, and how to think backwards from the clinic if you actually want to build something that matters. This episode is about what makes diagnostics really work. Not just the science, but everything that comes after: clinical utility, adoption, reimbursement, and survival. Welcome to the show this week, Arnon. How are you?
I'm well. Good morning to you.
Good morning. And where in the world are you calling from?
Cleveland, Ohio, if you couldn't guess from the name of the company, Cleveland Diagnostics.
Well, that's a great intro. We're yeah. So and spring must be upon us in Cleveland. It looks like the weather's quite nice behind you.
Well, kind of. This is actually what you're looking back is we reside in maybe 120-year-old brick factory that is now going through its third life, maybe, and you
Love
can
it.
get buildings like that for free where we live.
That's awesome.
Instead of
There we
trying
go.
to make them, architect trying to make them look like that anymore. But for us, I couldn't remove it if I wanted to.
Yeah, there we go. These places are all over the US, right? These buildings that need to
hmm
be revitalized. Well, look, we've got a lot to go through, and there's been, I think, since we've been in contact over the last 12 months, there's been some incredible news from you guys, and so I'm excited to get into it. I guess before we do that, it'd be great to get a bit of an intro to yourself, your background and career, and who you are.
So again, the name is Arnon Chait, and I'm actually a physicist. I would say by trade or by trade, I guess is a better way because I kind of changed careers many times, and I think I highly recommend it to anybody who sort of wants to have fun in life. And so I started up more in the engineering, went into physics, worked for NASA. And by the way, NASA did something big for the first time yesterday. It was a lot of fun watching it. But I spent there about 35 years or so doing all kinds of projects that I liked, and that was amazing. It's an amazing place. Mostly experiments in space and several other things that I cannot get into. But let's say that they also allowed me to pursue other hobbies at the time when we started any kind of the company, which is was put together by my co-founder Boris Aslavsky, who is a Russian biophysicist who just walked away in the early 90s, and we sort of found each other. He wrote a book, I read it, and we started discussing. So all I wanted to say is it allowed me to do it on a part-time basis, and then I left NASA when obviously the company grew up to a point where I couldn't do 5 jobs anymore. So my wife told me that. So
Yeah, good for you. And so, 30 years at NASA. I mean, what an incredible career in its own right. And then, how did the transition happen from NASA into diagnostics?
like anything else in life, most people, if you tell them, tell me what's your next 5-year plan, and they'll give you, and then 5 years later you ask them, it never matches. So I never had a 5-year plan. And whatever I'm interested at the time, I try to pursue that. And again, highly recommended. When I bumped into Boris, he was working on some bioanalytical technique that eventually led to the development of the core technology that we're pursuing. But then we worked on early stage drug discovery operation on other pieces, and then diagnostics actually came. We're asking the same question: is there information in proteins, which is what we study, that are in circulation that would be relevant to diagnostics applications? And that's how things just roll on their own, and then you get to diagnostics, and then we focused on it.
Amazing. And so, with such a rich history in NASA, the amount of experience that you've taken there. I'll probably never get the chance to ask this question ever again. So, is there
You may.
is there is there experience and sort of lessons learned and ways of doing things from NASA that you would take into building a diagnostics company?
That's a great question, actually. So of course you can say, well, no, they're two different businesses, but no, it's the mode of operation that is what you get soaked in when you're at NASA. And remember, it's a government agency, so it's a bureaucracy, and it's very attuned to the local politics of the day. Otherwise, they don't get a budget. So all of those things you say in a company, you don't have that. But how you actually put together a rocket or to put together a space experiment, so to put together all of those things that we so admire, be surprised and perhaps not surprised that it takes very few people who are super committed and super good and super smart to actually do it together. We all had our black books, kind of, if I want to know something about, I don't know, space plasma, I know who the guy to call. And so we have an organization that actually moves slowly the whole thing. And then we have these little pins, if you wish, that we all rely on each other to actually make the breakthroughs that could move the whole thing. So, and that works for diagnostics as well. I think it works for any company that is science rich. How do you push science while working with VCs or working with your reimbursement payers? Or, you need both at the same time, but your driving the innovation, that's common from large organization to small.
Mm. It's interesting, isn't it? A government agency, bureaucracy versus investors that you have to answer to. I'm
Same
sure
thing.
that, yeah, same thing. What did you feel like? This is my last question on NASA because I promised. But did you feel when you were at NASA? Because my assumption is that you're surrounded by some of the most intelligent people in the world, right? If you work for NASA, did it feel like that when you're there? Are you surrounded by people where you're just like that's the geniuses, legitimate geniuses everywhere?
If you have too many geniuses, nothing gets done. So and that's true for companies as well.
Right.
So you need a range of people. We need regular engineers who know how to engineer things and put together hardware and et cetera, et cetera. But then you need actually people who would say, all right, we need to get another 200 degrees on the rocket final stage there, otherwise it's going to melt. So make me a new material. I mean, they kind of push the envelope type things and there is a respect and the system is built around it. Incidentally, there is one, perhaps I'll throw a little piece. A lot of companies are claiming to do that, but NASA did that. When I retired from NASA, if my salary would have been on a full-time basis, I was only part-time at that time. It would have been, I think, $15,000 less than the NASA administrator, which is like way above me, right? And that's the whole idea, is recognizing that the top level in science at NASA, you don't need to go to management. Not everybody. As a matter of fact, you don't want to have your best people going to management. So they have a dual ladder that actually works, that respects the best people that they have with the same rewards that that the administration gets. And that's kind of an interesting thing that you can apply to companies. I'm trying to do it here as well. My best people should stay in science.
I think you've just raised two incredible points that are universal in that, that if you if you have great individual contributors, regardless of field, you want to keep those people as individual contributors, and that's
Yeah,
where they're going to be motivated. Um, but yeah, I mean, absolutely fascinating that too many geniuses don't get enough things done. I think there's some great lessons there applied to any type of science and scientific field. Um, so let's talk Cleveland Diagnostics. So
sure.
how
Yep.
did it come about, and take us back to the start?
So we had this little technology that I alluded to that Boris and I put together that actually asks a question, is proteins, obviously, we know that proteins really are the business of your life and disease. Everything is manifest in protein, not in genes, not in RNA, not methylation, not in whatever it is. It's manifested itself in protein because protein is, I mean, the lack of communication, for example, or too much communication or whatever changes to proteins are the key thing. But the technology that we did, and we thought initially about using it as a quality control for recombinant DNA, for example, products, which is to say, is there anything that changed in the structure of protein? Because the structure of protein is the final frontier, if you wish. This is where the business end of life is, the shape of proteins and changes to. So the question is, if you have a certain disease, is there a basis to think that there would be change to the structure of, say, some biomarker? And biomarkers don't have to cause the disease, but it could be modified by the disease process. So that's when we kind of realized that, yeah, you can pursue quality control, but if you can do that, then you can ask the same questions from a drop of blood to say, is there anything in there that has not been looked at yet that is connected to change to protein structure that can help a physician, reach a certain decision point? So with that business, when we start looking at it, the first test that we came to us, it's through a friend. They simply said, "Why don't you look at prostate cancer because it has such a bad name? PSA is a known protein. It's a great biomarker. Has a horrible name because it's not specific." So we ask ourselves, is there anything else about this molecule that we can look at with our technology that would get more specificity? And that's how we started this long journey. And it is long, and like anything else is life. Only the AI guys know how to do something fast. I mean, I don't know how to do it faster. Let's say it this way. In
And so,
medicine, it's slow.
when did you guys found the company, and sort of what's the journey been since that point?
We actually met our third co-founder, Mark Stowski, who is the MD in this picture. I'm just a PhD. Boris is a PhD, and we really needed a doctor. So he ran our first clinical study, and then when he moved to Cleveland Clinic, and which is perhaps one of the top hospitals in the country, academic institution, and he instigated with their venture arm and with the Urology Institute that let's put together, because this is a platform. We're not really stuck even to unique cancer. This we'll talk about it, I'm sure, more. Um, so we sat down with Cleveland Clinic and we put together their co-founding organization, and they gave us all the medical advice since then, oversight of clinical studies, some money. They've been wonderful partners.
So you've been working with Cleveland Clinic from
2013. Yes, you asked the one question that I didn't answer. Yes. Yep.
2013. Interesting. And so in that time, to take us through how the platform has developed and what you guys are doing now within prostate cancer. Take us up to today.
Yeah. So when you do anything new that nobody's used to, let's call it this way, and we didn't invent the fact that, for example, cancer modifies protein in specific ways. There are tons. There is literature. There are Nobel prizes been given to people who, in the past, solved protein structures. So everything is nice until you get to diagnostics, because we didn't have any technology that can do it within a clinical diagnostics framework, named something that is low cost, something that's going to be simple, that can run in the lab, and can tell a physician something new in not through a black box, but through some number that they can relate to, that relate to their experience. And when it comes to, even though we knew that structural proteins is changing, but we didn't know how to do it. That's all we did. If you want to put ourselves, what box did you put into? That's the box. We did a structural ELISA. So for those that remember ELISA, ability to look at concentration of any protein in the blood, let's call it. Yeah, we use that, but we do something in front of it that makes ELISA now reports on changes to the structure. So when you do, for example, we talk about our first product for prostate cancer. We'll talk about the clinical need in a moment, but the way it works is it asks essentially, is there a change to PSA that you see in the PSA molecule in blood that would tell you that it comes from cancer cells rather than benign cells, because ELISA simply says answer the question how much. We answer the question is where did you come from?
Mm. Okay. And so with prostate cancer and existing PSA testing, what you've mentioned the clinical challenges, the clinical problem. What are those clinical problems we have in prostate cancer at the moment?
They so obviously it's a number 2 cancer in men. It's if you think about it, it's akin to breast cancer in women. It's namely typically potentially is hormonally driven, et cetera, et cetera. But the main issue is that we do have PSA is a great biomarker for screening. Namely, in screening, you're trying to get high sensitivity, you're trying to catch something, but at the price of specificity. So you get, for example, if you have some inflammation, if you rode your bicycle to the phlebotomist, you will get elevation of PSA in your blood. There's nothing to do with cancer. So you bring a high level of PSA, and if you do it on an annual basis, your PCP doesn't know what to do with it, your primary care, so they send it on to urology. Urology doesn't know what to do with it because they don't have any more tools except for a biopsy, which is 12 needles through the wrong part of your body, et cetera, et cetera. You can think of sepsis. It's something that you cannot repeat. And a lot of men, for example, with benign conditions that enlarge their prostate would generate high PSA all the time. Well, you want to know if they have cancer. So how do you do that? And what we enter, for example, with the first indication of the same test, and it has many indications, potential indications, but it's to simply ask the question, does this high PSA come from cancer related to high-grade cancer or not?
I see. And so the high PSA level will mean that patients are being pushed through to biopsies unnecessarily.
Exactly. And that's you're touching on a very important point that I'm sure we'll get also is diagnostics is not one test and done. Diagnostics, in my book, it's always, they call it the diagnostics workup. It is one thing lead to the other, and you want to figure yourself where your technology fits so it can benefit the most and it would make the biggest impact clinically on the next step, on selection of the next step. Because typically as you go downstream, you get more and more complexity and you get higher cost and you get more comorbidities and you get everything that you don't want to. So, you have a good test upstream and and it fails in some things, focus on that piece. So then you can make an impact on what comes next.
Interesting. Does that mean the Cleveland Diagnostics test allows for, or the future is the the need for the PSA test is removed, or is the PSA test a first touch point that then moves into, okay, before we do anything about this, now we would perform the Cleveland Diagnostics test to make sure we're not pushing people unnecessarily through to an invasive biopsy procedure?
So it's the latter. You put it perfectly well. We could replace potentially screening PSA, but it is, when you look at the economics, it's much better to think of it as a two-step process, and we don't try to capture the whole universe.
Yeah.
We simply try, because we know that only about 10 to 15% of men screened by PSA would end up with high PSA. So why give it to the rest of it? But those 10 to 15% is still a sizable number, and that leads to sizable number of downstream biopsies and all of that.
Makes complete sense. And so the test itself, in terms of the development of the test, validation, commercialization, everything else, where are we in that journey, and what's the history of that journey of the prostate test?
It's actually the end stage, the truly end stage. And if you ask me, kind of, everybody's giving us kudos nowadays, and I'm kind of looking always back and simply say, what did I do again? Because we just did the thing that needed to be done without thinking that not too many people have achieved what you just said, this combination. Uh, And then I looked at data. It's interesting. For example, there are 10,000 publications about biomarkers for every one commercial product. This is an amazing piece. And then if you look at the attrition, why is that? It reminds me almost drug discovery, you know? I mean, there have even more when you start with CombiChem libraries. But it's, you know, if you really look at all the reasoning, you realize that most of them have to do with you can get a test, you can even show that it works, and then you start failing like big time. Like, for example, show that it makes clinical utility, show that physicians will use it, show that you have the gumptions to go through the, let's call it, I call them the lean years nicely. The years in which you think that you're going to go bankrupt every 5 minutes, when you have to give the test for free because the system here is so messed up that you have to prove that something works that has utility before you get reimbursement. We can get that's on and on those. If you get through all of that, and then convincing physicians, so you have to have people write lots of papers about it to show that it works and how it works with other modalities. I'm kind of amazed almost that people get through this. And clearly, I mean, David, if you're looking for another topic, is like, and I love this Diagnosing Dx. It's a great name because that is exactly the crux of the matter. Diagnostics spend 2 cents for every 2%. Yeah, so 2 cents for every dollar that is spent in healthcare, but they influence in cancer 100% of the decision, and all of healthcare about 60-70% of this. So it's kind of like the pain that we just don't understand. I mean, why is that? Well, we know that's a separate conversation.
That's a big conversation. That's a big
Yes, it is.
conversation. And
Let me join in on that later
I don't know
on
if
when
there's
you
an
do.
answer to it as well, Arnon. I just think it's a black hole conversation that never ends, unfortunately.
There is actually, and but that's again, and we can have it when we're done. We can have it on a beer, as they say.
Love it. Okay, we'll definitely do that. So test is now fully commercialized, and there was FDA approval fairly recently. Talk us through the significance of that.
Yeah, so we commercialized it initially and still do as an LDT, a lab developed test.
Yeah.
We have a clinical lab right upstairs. So the story behind LDT, because it's regulated by CMS, you can run it, and you have to use one lab that developed it. So in our case, it's in Cleveland. So you know that's the fundamental story behind it. But by the time you get to IVD, namely the FDA approved you, and it's not a single site, so you can sell actually kits. You can sell small little tubes that have our reagent that go in front of ELISA. And anybody who has a certain analyzer, immunoanalyzer that run PSA, can actually run our test. So they don't need us anymore. They don't need to send their blood. And that has a lot of advantages because they're literally, let's call it between 10 and 20,000 labs that can then immediately became a potential customer, and they can distribute the test much more efficiently than we can. So there are other benefits, of course, both on the financial side to the lab, but fundamentally it's a much better application of the technology that we have, which is so simple because it sits right in front of what exists in the lab.
And so a massive benefit of that is I would assume access, because you're starting to change the clinical workflow, and I assume would take physicians less convincing, you know, to adopt your test into their existing workflows, and you're trying to make that clinical workflow efficient and easy, and everything is all about increasing adoption.
Correct. And that's, if the test works, then everybody should use it, as far as I'm concerned. It's already shown utility. It shows benefit to payers, to the healthcare system, to physicians. So by the time you convince everybody that you have something really good, it's your job to make sure that everybody can use it. So, and that's really the piece that FDA approval, aside from the oversight and the rigor that they go by the process to approve, which was an amazing journey, I can tell you that.
Yeah.
But it's necessary. At the end of the day, you got this stamp of approval. People trust it, and your patient should trust it if you're a physician.
Yeah, that makes sense. And so we've talked about workflows and the clinical challenges. Did when you went into sort of the R&D and the early, development of the platform, did you think about the end clinical goal a lot as a priority? Was that something you had in mind about how you were going to impact the end goal? Was that part of the early stages of development?
I can tell you that today, when we work on additional pieces in our pipeline, which I'm sure you'll ask, we do that. We do this analysis ahead of time before we enter into anything. But I cannot tell you that we did it with the first product.
Right.
We simply ask ourselves, we know we can see changes to the structure of proteins. Somebody said, "Oh, PSA is a great example because it doesn't work well. Everybody knows it. Nobody uses it because of that. Can you fix it?" So we said, "Well, all right. Let's just see if we can see changes to the structure between cancer and non-cancer." And then the ball starts rolling.
Right. And so now you have this platform that is primed for different indications and tests, and you're now flipping that to go, okay, where do the biggest clinical problems exist that could be impacted by positive development?
Yes. Yeah, exactly. And again, by the time you get to that point, then there's a ton of considerations. How hard it is to get a specific claim from FDA, for example, that fits you into an indication. How big is that market for that indication? Who do you need to convince? What is the go-to market? I mean, who might be like the first adopters? There is so much stuff that goes in, which is, uh, you have to have somebody like Bob Rochelle, who is our chief commercial officer, who is a tireless expert in all the above. So I can put the test in front of him, and Amy Kastranik, and Mark Stovsky, who is our CMO, and we can do all of that. But if Bob doesn't do his job well, we'll never get there. But he did his job well.
So I guess I would love to say that's natural, but I don't think it is. I don't think enough diagnostics companies look at things that back to front way, but it is a recurring theme that whenever I'm in the fortunate position to speak to very successful diagnostics executives, they all seem to share that trait of understanding you have to look at things back to front, and you have to understand, is it going to be easy to implement? Is it going to be widely adopted? Are patients going to be able to or want to take the test and get access to it? That back to front thinking in diagnostics is, we see it here and there, but we always see it in really successful diagnostics executives.
I think that not you touched on the most important piece, because when we talk to the bankers and other kind of Wall Street types and whatever analysts, equity analysts, etc., and I always ask them, even though I know the answer perhaps, and they all
Yeah.
say, there are many companies with great ideas and great data, but not too many actually have done the A to Z story successfully. And I cannot tell you that it's all, we are so smart when we started it that we understood that. It's kind of much more organically,
Yeah.
and I guess with some luck also works. And you have to have a test that works, actually, by the way. So,
Yeah.
but that is exactly right. And I envy my drug companies colleagues, because in their case, typically a drug in the US is a binary case. The trial works or it doesn't work. If it works, you're in the money. If it
Hmm.
doesn't work, you fold and you move to somewhere else. I mean, that's how the biotech story works.
Yeah.
For us, it's always the endless inquisition from all these buddies that gets you to the end. So I'm always thinking it's much harder, actually.
Yeah, that's really true. I've never heard it explained like that before, but it makes complete sense. Diagnostics is full of great science. Doesn't mean there's going to be great impact or commercial success, and
Hmm.
that's fundamentally different. I think that's really well put. So what's going on today, Arnon? Where's Cleveland Diagnostics and yourself, and what does the future look like?
So with regarding to the test that we have, ISO PSA, as we said, well, we proved all the points around it, including the coveted FDA approval. So it's our job actually to make it available to everybody. So
Mhm.
we're really focused on commercialization. We have some new excellent assets, in management and others. We are working anywhere from compliance to quality to manufacturing to all the little details, by the way, that drug companies maybe don't have to. Maybe they do. So we are really busy staffing up and attending to everything so we can actually distribute the kits effectively throughout everyone. And that is it's going to take us years, no question about it. But that's how you do something that becomes the standard of care also. It
Yeah.
takes all of those things. In the future, simultaneously with that, I alluded to the fact that this technology is really platform,
Yep.
which is depending on who is the listener, it's either a curse word or a recipe for success or a recipe for failure, or again, depending who is the customer here. But we proved it. So ISO PSA for us is a proof that the technology works. So as you're asking me, is like, what else is there? Well, we have a lot of other cancers of interest, right? They all have, and if I said generically that all cancers modify proteins, then your question to me is, okay, then what next? And what's the biomarker? So we do, I mean, we have data on multiple interesting, and typically we work in research on multiple potential projects. And then remember, you asked and you answered that you may start and stop project, perhaps has nothing to do with the decision has nothing to do with performance. It could well
Mm,
be maybe the market is too small, or there is a competitor, or you can't get enough money out for it, or whatever it is. So,
yeah. And so, and how about yourself, Arnon? Because you've
mm,
recently changed your
yeah.
capacity in the company, and were previously president and CEO. So what's changed now, and how does that impact where the company goes, and in line with those plans?
So by the time we got to our stage, which is north of 100 people, then you have really a regular company. Surprise, surprise, you wake up one morning and then you have a company. And that simply means that you have all the commercialization, and then you have the company structure and HR and everything under the side. And the commercialization, we have to really rapidly commercialize and at the same time develop something new. So it's really multiple companies in one. And it occurred to the board and to me that uh, like it or not, this is you cannot the job of a CEO, if you think about it. Yes, even though it's to oversee the entire company, but we should really pay full attention to the commercial part. And there are other people that have more experience than I do. At the same time, we know that the value proposition is tied to the platform. It's tied to our ability to generate test after test. We know how to do it. We know how to get it through regulatory bodies. We know how to commercialize stuff. So keep doing it. So somebody has to take care of that. So that's where we split the job, if you wish. So Mike is our new CEO, Mike Iskra, who has had tons of experience commercializing, scaling up, doing all the good things. We work hand by hand in actually doing this entire enchilada together.
So that makes sense. So actually, so it almost feels like you're going back to the startup of what you previously did with on the prostate test, and now it's about bringing the new tests on the platform, and you're basically going to be leading that charge and developing the next functionality to bring to market. Is that something that you feel more passionate about as well? You think you'll enjoy that part of it again?
Yeah, clearly, as you started up, I mean, I started up as a scientist and I'll die as a scientist,
Yeah.
so there is a lot of stuff in between. But yeah, you're absolutely right. The only difference is I want to make it much faster. I mean, we have very ambitious milestones for the research component because we know that it works for a change. And then we know that because the other side is super hungry, namely the commercial side, regulatory, manufacturing, everything, feed them with new stuff. And
Mm.
so we want to actually change or provide, let's call it, the entire field of diagnostics with access to information that we know is there, but has never been touched. So because the test is sufficiently, all tests runs similarly. They use very inexpensive instruments that run at every lab. And if they can aid in different indications, they will be in a different position along this diagnostics workup. But if they can provide useful information, then diagnostics would, as a field, would move up. So instead of just having ELISA, you'll have ELISA and then something else beyond that would give more information for the right case.
Yeah. And so what's changed in the last 12, 13 years since you guys started Cleveland Diagnostics compared to today in the market? Do you think it's easier to bring new diagnostics to market today or more difficult? How's the landscape changed in your opinion?
Uh, Especially diagnostics, more than drugs and more than procedure thing, is so tied up to technology. So you give us a new box, like Illumina's or whatever, sequencer or whatever.
Yep.
We start asking, the questions of what can we solve with this box? So the technology typically drives the development of diagnostics fundamentally, which is, so we hope, for example, that what we develop in the future would be in the same way. Now you have access to different type of information. So liquid biopsies, for example, can answer a lot of questions extremely well. Like, is this EGFR mutation and drug combination would work? That type of question you can. But it's not really that great when it comes down to say, you have high PSA, is it coming from cancer or not? Because that has to work on a larger population, has to be low cost sufficiently, much more than sequencing. And it also cannot be black box that combine a lot of things. It has to be simple. There's a lot of attributes as you go upstream in a disease. And that's what we can fit in quite nicely, I think. I kind of went meandering around the question, but that's essentially what changed, if you wish, for us. The other little thing that obviously I have to throw in is AI, and it's AI would change a lot of things. We are also quite interested in let's call it converting what we do, aside from the ability to measure changes to the structure, to measure changes to the structure in the context of an individual. So you can ask yourself, can AI actually incorporate everything that you know about that patient together with your information, and then provide a much more actionable and in an easy way to the physician. So that's how you can make an impact on medicine. How it's going to work, I do not know, but it's definitely coming.
Do you think that allows for more easily repeatable not constant, but screening of potentially high risk patients in that could
Yeah.
be one of the biggest benefits? Yeah.
Yes, absolutely. So it's almost kind of like you wanted to say, I kind of feel bad sometimes. The last trial that Grail had in the UK,
Yeah.
they had to rely on only the methylation signatures, to say we succeeded or didn't succeed, and that up to the regulatory bodies and the payers and everything else. What would have happened if, for example, all information regarding those patients would have been available upfront to Grail when they did this huge study? Could have developed something, and perhaps they still could, that would make their technology works best. We certainly are interested in pursuing something like that.
Yeah. And so what's your objective outlook on the diagnostics market as we see it at the moment, Arnon? Where do you think we are as an industry?
I'm super bullish. And people will say, okay, well, all right, then invest in the stock market or something on this. No, I'm saying it because it's kind of maybe the wrong reason, because of the failure especially in the US, but even in the UK and other countries who actually embrace a single payer, uh, they uh, we cannot afford the healthcare that our technologies, our drug and procedures and everything else are allowed. So it's almost like if you're saying, if what I love the concept that for example, NHS invented the quality adjusted life year, which actually say we as a society have only finite amount of resources. And if you give us something that costs $1 million and extends somebody's life by a month, we're not going to do that as a society. And that is absolutely correct because we do it in every aspect of life, but we don't do it in healthcare. God forbid you do it in healthcare, especially in the US. That that story is about to end. So that would, as far as I'm concerned, we're there already. You cannot afford all the super exotic stuff that we're developing today. So the question is, who should get it? And not only that, reduce the number of people who need that right up front. Do things that are much simpler. If I can tell you today that you don't need a biopsy, I saved $2,000 to the healthcare system, and a lot of anxiety and potential some death or whatever it is. It might be due to sepsis and who knows what. But if I can do it for many other indications, that would simply make the healthcare system more affordable. So, in the U.S., the whole buzzword today is on prevention, nutrition, etc. You can only prevent so much. People are going to die, and people are going to die from diseases. So the question is, how can we afford as a country, all the goodies that we can develop? And that's why I think diagnostics is the solution.
What do you think needs to change to push the industry forward in terms of actually becoming the solution in clinical workflows?
I think that we are internally just the metrics of Wall Street and very large. I mean, they're such that the industry's head, like everybody else, is in the sand. We cannot think of anything. I have to get my sales up. I have to get another test out. Whatever it is. The advocacy for that's why I'm kind of saying, well, I'm quite optimistic because uh, it's on the congressional level in the U.S. People would realize that healthcare system in the U.S. is a $5 trillion organization. It's expense. It's a heck of a lot more than going to wars. It's a heck of a lot more than everything. So we are only 2%. Give us 3%, and I'll change the world.
Yeah. Arnon, thank you so much for coming on and sharing the story, because I think there's been some incredible publicity. The Cleveland Diagnostics name is now, it's obviously it's really well known around the industry, and obviously with the great news that you guys have had in the last 12 months, especially around the FDA approval, there's a path there that companies should look to follow. But it's also interesting to hear from the inside about how you intend to do things differently as you expand the platform and faster, and obviously sort of thinking about things in a back to front way now, because you can. What lessons can be taken there into the early diagnostic single product startup that's out there now, who's thinking their science is great and actually really needs to start thinking bigger picture, what's going on in the clinic. It's fantastic to hear from you, and just thank you for coming on and sharing the story and your own perspective on the market. Fantastic to hear.
And it's been my pleasure. Everybody likes to talk about things that are dear to their hearts, and I hope that our conversation would benefit somebody. I agree. And I'm always available, by the way, to tell you what not to do. I cannot tell you too many things how to do it, but I can tell you about failures or shortcomings or things that I should have done. And yeah, somebody wants to contact me, be my guest. This is great.
The lessons learned are there. That's what we need to
Yeah.
take from these things. So thank you very much, Arnon. Thank you for coming on.